Welcome to the Cosmos Web Portal tutorial. This video will show how you can execute the virtual cell-based assay developed for the web portal. The virtual cell-based assay consists of ordinary differential equations represented by 4 sub-models: a fate model, a cell growth model, and a toxic effect model. The purpose of the tool is to simulate the fate of a chemical and the intracellular concentration leading to the effect and to cell perturbation. For more information, I would like to encourage you to download the relevant documentation from the Cosmos space. We can see here on the left the KNIME workflow that was developed to include the virtual cell-based assay, and on the right side a table summarizing the chemicals for which now the virtual cell-based assay web portal version can simulate, and the different type of cell lines in single and repeated exposure. So now I would like to move to the Cosmos KNIME web portal for a short click-through session. In order to access the Cosmos KNIME web portal, you need to register and to get your access detail. Then once you logged in, that the main page is divided in 2 parts, and by selecting the repository file public, you can then access under the biokinetic file the virtual cell-based assay model. By clicking on the virtual cell-based assay, the VCBA, you can see on the right side that a short description of the model will pop out, and then you can click start, and you will have the possibility to input some parameters, so to choose which type of cell line you would like to simulate, which type of chemical, for instance today caffeine, which type of exposure, single, repeated, or you can also try to optimize the parameter or validate your model. You need to input also the number of cells, the time of your incubation, so usually 48 hours or 24. You can select the assay volume which you used in your in vitro testing, and if you are in the repeated exposure mode, you can also have to input these last 3 parameters, such as the initial concentration, the interval time, and the number of exposures. Finally, you must introduce the concentration used in your in vitro testing. Then you press next. You wait that the workflow will execute. It will take around 5 minutes. So here you have displayed the results, and you will see that you will have 4 types of graphical outputs, and these graphical outputs also will be available in the report version, which you can download as a PDF, a doc, or any other type of file. So with this, I would like to thank you for listening and watching our web tutorial.